The overall goal of this procedure is to perform imaging and analysis of neural progenitor mitosis in live mouse brain slices. This is accomplished by first dissecting brains from embryos. The second step is to prepare brain slices with a vibratome. Next, the live imaging of brain slices is performed. The final step is to extract information from movies to analyze mitosis parameters of interest. Ultimately, time-lapse microscopy is used to show how progenitor populations of the brain divide in real time.
The main advantage of this technique over existing methods, such as fixed analysis, is that one can gain significant insight into temporal dynamics of neural progenitor division in live brain slices. It can also be used to understand how genetic perturbations may influence neural progenitor division. Though we use this method to understand cortical development, the analysis we described can be applied to the study of mitosis of stem cells in other brain regions, or even outside the brain. Demonstrating this procedure will be Louis Yan, a postdoctoral fellow from my laboratory.
Begin this procedure by collecting the mouse embryos and dissect out the embryonic brains, including the hindbrains and the forebrains, with the two cerebral hemispheres. In a bucket of ice, create a hole in the ice for the embedding molds. Next, pour 3% agarose medium into a plastic mold and place that mold into the hole in the ice. Stir the agarose medium with the tip of a digital thermometer until the temperature reaches 35 degrees Celsius. Then, promptly transfer the brain into the embedding medium. To remove excess HBSS at the interface between the brain and the agarose,